During ejaculation, nearly 300 million sperms move from the testes through the tubes called the vas deferens into the area of the prostate. At this point, the prostate contracts, closing off the opening between the bladder and the urethra, releasing fluid into the urethra and pushing semen on through.
Ovulation is a part of the menstrual cycle when the ovary releases a ripe egg, or ovum. Inside the ovary are hundreds of thousands of follicles. Each follicle is a hollow ball of cells with an immature egg in the center. The typical 28-day menstrual cycle begins on the first day of menstrual bleeding. During the first 7 days of the cycle, a few follicles begin to grow at the same time. These maturing follicles secrete estrogen hormone into the bloodstream to prepare the lining of the uterus for pregnancy. Around day 7, all of the follicles stop growing and begin to degenerate, except for one. This dominant follicle continues to grow and nourishes the developing egg inside it. Around day 12, the follicle secretes a large amount of estrogen into the bloodstream. When the estrogen reaches the hypothalamus and the pituitary gland in the brain, the anterior or front part of the pituitary gland releases a huge surge of luteinizing hormone into the bloodstream. Around day 14, luteinizing hormone causes the follicle to undergo a sudden growth spurt. Right before ovulation, the egg detaches from the inside of the follicle. The bulging follicle releases chemicals, causing one of the two fallopian tubes to move in closer and surround the follicle. The follicle swells until it bursts open, ejecting the egg and fluid from the follicle into the abdominal cavity. In response, the fimbriae, tiny projections at the end of the fallopian tube, sweep across the ovulation site and pick up the egg. Microscopic cilia on the fimbriae surface transport the egg to the entrance of the fallopian tube. Inside the walls of the fallopian tube, muscular contractions gently push the egg towards the uterus.
First trimester surgical abortion called suction D&C, dilatation and curettage. This is the most frequently performed abortion and is used typically from 5 to 13 weeks of pregnancy. After administering anesthesia, the abortionist uses a speculum like this. This is placed inside the vagina and opened using this screw on the side, allowing the abortionist to see the cervix, the entrance to the uterus. The cervix acts as a gate that stays closed for the duration of pregnancy, protecting the baby until it is ready for birth. The abortionist uses a series of metal rods called dilators, like these, which increase in thickness, and inserts them into the cervix to dilate it, gaining access to the inside of the uterus where the baby resides. The baby has a heartbeat, fingers, toes, arms, and legs, but its bones are still weak and fragile. The abortionist takes a suction catheter like this one. This is a 14 French suction catheter. It's clear plastic, about 9 inches long, and it has a hole through the center. It is inserted through the cervix into the uterus. The suction machine is then turned on with a force 10 to 20 times more powerful than your household vacuum cleaner. The baby is rapidly torn apart by the force of the suction and squeezed through this tubing down into the suction machine, followed by the placenta. Though the uterus is mostly emptied at this point, one of the risks of a suction D&C is incomplete abortion. Essentially, pieces of the baby or placenta left behind. This can lead to infection or bleeding. In an attempt to prevent this, the abortionist uses a curette to scrape a lining of the uterus. A curette is basically a long-handled curved blade. Once the uterus is empty, the speculum is removed and the abortion is complete. The risks of suction D&C include perforation or laceration of the uterus or cervix, potentially damaging intestine, bladder, and nearby blood vessels, hemorrhage, infection, and in rare instances, even death. Future pregnancies are also at a greater risk for loss or premature delivery due to abortion-related trauma and injury to the cervix.
Bill, there are two types of vacuum aspiration abortions. The first is manual vacuum aspiration. The second is machine vacuum aspiration. To perform a manual vacuum aspiration abortion, also called an MVA abortion, an abortionist uses the suction of a syringe to remove the baby from the mother's uterus. This method of abortion is not available everywhere and may be more common in poorer countries. According to the Grantham Collection, the major problem with the MVA is that it has a very weak vacuum. This means that the child is ripped apart slowly. A machine vacuum aspiration abortion is performed in a similar way. This method uses a small tube called a cannula. This tube is inserted into the uterus and attached to a pump. When the pump is turned on, the child is torn apart and pulled through the tube into a bottle.
The water is churning with chemicals and organic compounds. Lightning strikes the chemical soup at the right place and the right time. Billions of volts of electricity trigger a chain of improbable coincidences. The chemicals' atoms join up in a precise sequence, creating a bundle of genetic material. These fragile genes don't stand a chance in this extreme environment. But luck strikes again. A blob of oily material engulfs a single chain to create the first ever cell. Now, the genes send out messages, chemical instructions. And 3.5 billion years ago, they do something extraordinary. They copy themselves and the cell to create a perfect clone. This is the very first living thing. Every human, every animal, every bug, every plant can trace its origins to this single cell. The genes tell each cell to reproduce, guaranteeing their survival as they pass from one generation to the next. For 2 billion years, the only living things are simple single cells, but a random accident changes everything. 2 cells merge. Their genes combine. The merged cell clones itself. Its offspring contains genes from not 1, but 2 cells, 2 parents. We call this accident sex. Sex introduces variation. Occasionally, things go wrong. As the cells reproduce, genes get deleted, duplicated. These cells are mutants. Mutations pile up, differences increase, until the cells become so different, they're separate species. The tree of life branches out into billions of species, but only one will lead to us. Mutating and diversifying, spreading out through the oceans, getting bigger, more complex, until our ancestor is a 3-inch long water worm. This is us 550 million years ago. we leave solid ground for a new life in the trees. A new world demands a new body. We've come a long way from that first single cell. Now, at last, we're beginning to glimpse a family resemblance. We're Altiatlasius, one of the first members of a new group of mammals, the primates. By 56 million years ago, we've adapted to our world. But that world is about to change again. Over 10 million years, extreme temperature shifts ravage the forests. Forests get smaller, food gets harder to reach. Natural selection intervenes again. Our tail shrinks back to the base of the spine, where it stays, as the coccyx, a reminder of our days spent leaping through the trees. Instead of leaping, we stretch. Our arms grow long and flexible. Changing temperatures changed our ancestors, and us, forever. But the planet's not finished yet. Deep underground, the African and Arabian plates pull apart. The land between them drops to form the East African Rift Valley. Along its edge, a 3,500-mile-long mountain chain rises up. Rains from the Indian Ocean once watered all of Africa. Now the mountains block their path. The trees get even further apart, the food harder to reach. We're starving, desperate. 4.4 million years ago, the will to survive that's driven us from a single cell in the ocean to a complex primate in the forests of Africa is about to make us do something extraordinary. Something no primate has ever done before. After 3 billion years of evolution, we hit an evolutionary dead end. We're Ardipithecus ramidus, 4 feet tall, at 80 pounds, with a brain the size of an orange. Our dense rainforest home has become patchy forest. We can stand on branches, move along them. But to reach more food, we need to let go. .4 million years ago. These are our first steps on 2 legs. Walking takes us to the food and leaves our hands free to pick it. It catches on, fast. Ardipithecus is smart enough to copy and learn. Walking gets passed from parent to child. Over the next 1.2 million years, our body evolves so that we can walk further and faster.
Hey smart people, Joe here. Charles Darwin spent 5 years circling the world to discover what became the origin of species. Alfred Russel Wallace spent 8 years traveling through South Asia to witness the story of evolution. But you can discover the same thing today without going anywhere. Because the story of evolution is right there in your body. Your tailbone used to be a tail. If you can move your ears, that's thanks to muscles you share with other mammals. That pink bit in the corner of your eye, a leftover of a third eyelid we can still find in lizards, birds, fish, and others. Goosebumps are a feature we share with lots of animals that didn't lose their body hair. And even hiccups trace back to the brains of fish and tadpoles, a brain signal they use to squeeze water through their gills. These body parts are just a few scattered pages in a larger story about our evolution. But there's other fossils hidden inside our bodies that tell an even clearer tale about how we came to be the way we are. One of these stories is so simple it can be told with a single picture. These are images of chromosomes, the big chunks of DNA inside every one of our cells. When we look at images of human chromosomes next to those of other apes, they line up nearly identically, except for human chromosome 2. It seems to have no match next to the other ape chromosomes. Even weirder, humans have 46 chromosomes in total, 23 pairs. Those other apes, they have 48 chromosomes, 24 pairs. What could explain this? If we're supposed to be related, why are we missing a chromosome? Turns out we aren't missing a chromosome at all. Sometime during our species history, those 2 chromosomes that are still separate in other apes were welded to become our single human chromosome 2. Us, chimps, gorillas, orangutans, all inherited the DNA we have today from some common ancestor with 48 chromosomes. But 2 of ours got stuck together along the way, and you can even see how they match up. If 2 chromosomes were glued together, you'd expect to find 2 centers, or centromeres, and we do. We'd also expect to find telomeres, the ends of each original chromosome, stuck head to head in the middle, and we do. Thanks to sequencing the human genome, we can spot the exact DNA base where this fusion happened. 113,602,928 bases from the end. A fossil of evolution hidden in our own bodies. In Celtic mythology, there's a place called Tir Nanog, a land of eternal youth. When one scientist discovered a protein that keeps stem cells forever young, he named it after this place, Nanog. The human Nanog gene is located on chromosome 12, at least the version of the gene that works. There are 11 other broken copies of Nanog spread throughout our DNA. How do these extra copies get spread through our genome like lost socks? When DNA is replicated, sometimes the same bit gets mistakenly copied twice. And sure enough, we see one broken copy right next to the working Nanog gene, like a messed up DNA photocopy. We call these broken genes pseudogenes. And what about those other 10? There's another way these pseudogenes can spread. Nanog, like every gene, is copied from DNA into RNA letter by letter before that RNA is used to build a protein. But every once in a very long while, that RNA gets mistakenly copied backwards back into DNA, pasting a new broken version of the gene somewhere in our chromosomes. After searching the billions of letters in our genome, we've found 10 of these lonely Nanog leftovers in our DNA. Their locations seem random, like they were blindly tossed in. Now, say you were to read another animal's genome and you found these very same broken copies of Nanog, each pasted in the very same locations among billions of letters of DNA. That would be a heck of a coincidence, unless it wasn't a coincidence. This is what we see when we search for Nanog in a chimpanzee's genome. The original and 10 of those lonely broken copies, all in the same locations as ours. This only makes sense if millions of years ago, you and me and the chimpanzee share an ancestor whose DNA was already littered with these broken genes. We all inherited this pattern because we share an evolutionary link. Of course, it's one thing to share a link with chimps, but chickens? That would require some strong cluck and proof. One thing you have in common with a chicken, you both came from an egg. Inside a chicken egg, the growing embryo produces something called a yolk sac. It surrounds, well, the yolk and helps gradually absorb nutrients to build a baby bird. But human eggs are incredibly small, only about 1/10 of a millimeter across. Unlike birds or turtles, our eggs don't make a yolk or even a shell. We get our nutrients and protection from our mom's body. So you'd think no yolk sac. But that's not the case. Humans make an empty yolk sac, and it disappears halfway through pregnancy. See, this is a clue that our eggs and bird eggs and even reptile eggs all share an evolutionary link. But can we prove it? There's an essential ingredient in egg yolks called vitellogenin. It's a protein, which means there's a gene whose DNA codes how to build it. Egg-laying vertebrates all have at least one copy of this gene. But what about us? If we really are descended from animals that once laid eggs full of yolk, can we find some leftover of that yolky gene hidden in human DNA? In 2008, researchers found just that. Fragments of once active egg yolk genes hidden in our own genome. And when they compared our DNA to the chicken versions, they even had the same neighboring genes around them. This is exactly what evolution would predict. Just like fossil bones, we see remnants of our ancestors broken and buried in these hard to find places. There are many myths of creation around the world, and most agree on one thing. Humans are special, and we need a special story for how we got this way. When we take a close look at our own biology, we can see that special story is evolution, and it's a story we share with every other living thing on Earth, some more than others. To some people, the idea that humans, our bodies, our brains, our thoughts, are also the products of evolution removes some of the magic of being human, but it doesn't have to. When Darwin wrote, "There is grandeur in this view of life," he meant that being a creature molded by evolution is a totally different kind of magic. The kind that still amazes you even when you know exactly how the magic is done. Stay curious.
When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their acrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, their outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed male genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm with the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads, called microtubules, pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she will implant in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.